Astrocytes are these star-shaped cells. That's why they're called astrocytes. They're really important in bridging connections between the blood and the nervous tissue. They help to form a really important structure called the blood-brain barrier. This is a bridge or a filter somewhat that protects the nervous tissue from all of the gunk that's traveling in the blood, right? The blood is exposed to toxins and drugs from medication, and pathogens, bacteria, viruses are traveling in the blood. And we really don't want a lot of that toxic material to freely communicate to our neurons, to freely move into the nervous tissue. So astrocytes have these finger-like projections that stick out like a star, and they bridge from the neuron to the blood vessels, and they prevent the transmission of any of that toxic material from traveling from the blood out into the nervous tissue. They're also involved in synaptic modulation. So what we just spoke about, synaptic facilitation and synaptic inhibition. And as we can see here, this exoaxonic connection here between the axon of the astrocyte and the axon of the neuron that it's modulating facilitates that synaptic modulation. Astrocytes also communicate through chemical messengers, but again, they are non-excitable, so they do not have or transmit action potentials. They help to clean up the environment, so they remove any excess neurotransmitters. We talked about the importance of clearing the synaptic junction, clearing the synaptic cleft. After that signal has been sent, and astrocytes can come and help clearing those excess neurotransmitters from the junction. They also maintain normal electrolyte composition of the interstitial fluid in the CNS. So basically keeping the amount of sodium stable, the amount of potassium specifically stable, the amount of chloride stable in the interstitial fluid that's bathing our neurons. And it's really important to keep those ions at stable concentrations, because remember, they are involved in actually firing an action potential. If we don't have enough sodium present in the extracellular fluid, then when we open up those voltage-gated sodium channels, we won't have, it will be harder to get to threshold, it'll be harder to fire an action potential. They also protect neurons against toxic substances and oxidative stress, as I mentioned earlier, by bridging that connection with those star-like projections. And then lastly here, they are involved in glycogen synthesis. Now the brain typically cannot store glycogen, but astrocytes do have the ability to store a small amount of glycogen. That's going to be really, really important. The brain can only use one source of fuel. It can only metabolize glucose. So we spoke earlier about cellular respiration and about the different biomolecules that can be metabolized to produce ATP. We talked about glucose. We talked about lipids, we talked about proteins. In rare instances, the brain is kind of exempt from all of that. It can only metabolize glucose. And so having the ability within these astrocytes to store some glycogen that can be converted to glucose is really, really important for sustaining the brain in the instant of starvation. And then microglia cells are basically the immune cells of the central nervous system. They function in collaboration with astrocytes to protect neurons from toxic, harmful substances. They also have this phagocytic ability. So they have the ability to engulf foreign particles. If they do make their way into the nervous tissue, they can clean up bacteria or viruses or dead, injured cells or any other type of debris that exists in the nervous tissue. And they also protect from oxidative stress, right? That free radical stress from the mitochondria. Okay, we can speak about some clinical connections with these glial cells. The last two cells that were involved are myelin cells, and I'll talk about those later on in this packet. But when we lose the ability to myelinate cells, we end up with a disease called multiple sclerosis. So this involves many different organ systems because it involves the demyelination of nerves. So movement is affected, speaking is affected, visual and hearing perception is affected with multiple sclerosis. We can also speak about two really common neurodegenerative diseases, Alzheimer's and Parkinson's. I'm sure many of you have heard of this disease or know someone who has suffered with this disease or these diseases. And they're basically a loss of function of specific neurons in the nervous system. So with Alzheimer's, we have loss of cholinergic neurons. That is neurons that secrete acetylcholine. And that is replaced by plaque or scar tissue in the brain. That usually presents as cognitive decline, so loss in memory, loss in other cognitive functions like judgment and reasoning skills. And then we've got Parkinson's, which is a loss of dopaminergic neurons. This is more of an inflammatory neurodegenerative disease. And Parkinson's kind of differs from Alzheimer's in that there's a movement component or a movement dysfunction associated with Parkinson's. So Parkinson's patients usually have that slowed gait because of the involvement of dopamine with movement in the brain. Okay, so we've got Parkinson's and Alzheimer's, which are two forms of neurocognitive or neurodegenerative diseases. Okay, so our second objective here is to describe physical structures that support the CNS. Now, a lot of these structures we've spoken about in our unit in the lab. So we talked about the skull, which includes the cranium and the vertebral column. Both of these help to protect the brain and spinal cord. And so they're hard bones that encase these fragile brain tissues and spinal cord tissues. We've also got the meninges. We talk about the three layers of the meninges, the dura mater, the arachnoid mater, and the pia mater, which wrap around the brain and spinal cord. And they not only protect, but they help to circulate that cerebrospinal fluid that cushions these substances from the hard structures that are the bones, right? And we can look at these structures here. We've got the skull, which is the outer bone that protects the brain. We've got the meninges, which is really formed by these three layers. The dura, that's thicker, tougher layer. The arachnoid, which is the intermediate layer. And then the softer, more delicate pia mater, which is closely connected to the brain. And these layers continue right around the spinal cord as well. Another part of supporting the CNS is cerebrospinal fluid, or CSF. This is produced in the cerebral ventricle. So we talked about the ventricular system in labs. The ependymal cells, which form the lining of the cavities of the brain, produces CSF. And that is a clear watery fluid which circulates around the CSF, specifically between two of those meningeal layers. So CSF travels in the subarachnoid space, which is basically above the pia mater but below the arachnoid mater. So in between the pia and the arachnoid, we've got the CSF that is circulating, protecting and cushioning the brain and spinal cord. So it's made by ependymal cells in the choroid plexus, and it circulates through the ventricles, and eventually it goes to the subarachnoid space, and it is reabsorbed by the arachnoid villi. So let's jump back to this image here. So here we can see the arachnoid villi, which are these projections up into these venous sinuses. This is where the venous blood of the brain gathers. And so basically, CSF is reabsorbed into the venous system, and so it's constantly being secreted by the choroid plexus and reabsorbed by the arachnoid villus. Okay. And as I mentioned earlier, its function is really cushioning and protecting and keeping a stable interstitial fluid environment in the surrounding cerebrospinal, in the surrounding central nervous tissue. Now, this table compares plasma to CSF. And what I really want to point out here is the different proportions of certain substances that's found in plasma and CSF. So there's a lot of proteins present in plasma and very little protein in the CSF. Likewise, glucose. So we've got lots of glucose in plasma, but very little glucose in the CSF. And that's partially because of the blood-brain barrier. So we'll speak about the structures that form the blood-brain barrier. but basically it is a size selective membrane. So large particles like glucose and proteins cannot really move across that barrier. And so we see them in high amounts in the plasma, but in very small amounts in the CSF. Alrighty. Okay, and then the last thing we'll leave off here with is just reviewing the ventricular system. So we talked about this at length in lab, but we know about the ventricles where the CSF circulates, these chambers within the brain, the lateral ventricles, the third ventricle, the fourth ventricle, and then eventually through the central canal, this CSF will circulate right around the spinal cord and make its way right back to those arachnoid villi, where it is then reabsorbed into the venous blood via these sinuses, and then that can be circulated continuously